Dear students, welcome to ST403, Statistics for Bioinformatics class. This is a 2-credit course which is not compulsory for either Statistics Honors students or SOR students. The aim of this course is to provide you an introduction to major statistical methods used in the field of bioinformatics. Recent developments in high-throughput technologies have led to explosive growth in the understanding of biological data, for an example, an organism's DNA, functions, and interactions of biomolecules such as proteins. Also, there is an increase in the databases that require new tools to store, analyze, and interpret the information contained in the databases. Hence, statistical methods can facilitate to achieve these goals. First of all, let's look at the assessment criteria for this course. There will be tutorials, quizzes, one practical assignment and the end semester examination. The tutorials and quizzes will be assigned online, unless the university reopens. I will be flexible about these percentages based on the performance of you all. In addition to the lecture note that I will be discussing, I will be posting helpful reading materials. Each week, a time slot will be reserved to have an online discussion to go through the topics that you need more clarification. Let's see what is bioinformatics. Based on perspectives, we can define them in different ways. Someone who worries about the size of the biological data could define bioinformatics as a data-related problem, that is representation of data, storing and retrieval of data and analysis, while another one may consider as a field of study in biological problems. Before we move on to statistics applications in biology, it is good to have an understanding on basic biology. Let's see what is genetics. Genetics is a branch of biology and plays the central role of biology. It is necessary to understand genetics to get understanding about biological data. Genetics is the study of heredity. Heredity is a biological process whereby a parent passes certain genes onto their children or offspring. The first scientific experiment of heredity goes back to 1860s, where Mendel studied how the characteristics of pea plants passed from one generation to the other. As I mentioned at the beginning, recent development in technology have led to explosive growth in data. These data are useful in understanding an organism's DNA. Now, you may be wondering, what is an organism? An organism can be considered as any living thing. These databases contain complex, noisy, and probabilistic information that needs adaptation of classical and non-classical statistical methods to new conditions. The new science of statistical bioinformatics aims to modify available classical and non-classical statistical methods to develop new methodologies and analyze the databases to improve the understanding of the complex biological phenomena. What we have discussed earlier could be summarized using this figure. Moreover, the research in statistical bioinformatics is advancing in all directions, from the development of new methods, modification of available methods, combination of several interdisciplinary tools, to applications of statistical methods to high-throughput data. When one looks at the growth in the number of research papers in statistical bioinformatics published in leading statistical journals, there are a number of fields that are getting benefits from the developments in statistical bioinformatics. This includes identify new genes and to provide knowledge of their functioning under different conditions. Learn about new diseases, such as cancer and heart diseases. Identifying the patterns of gene activities lead to classification of diseases according to their genetic profile, which would be an added advantage to the companies to find medication that target directly on the gene-specific or tumor-specific. Identify the relationship among drugs and genetic profiles. This could be done by studying the correlation between toxic ants and the changes in the genetic profiles of the cells exposed to such toxic ants. Here are few examples. The top left figure shows the normal rice, which is in white color, compared with golden rice. Golden rice is a genetically modified variety of normal rice. In the middle, you can see a picture of different tomatoes. The first block of the picture shows what happens to normal tomatoes over a time period of 45 days. You can see that the ordinary ones have become soften and shrivel up with the time. But when the gene that accelerate this shrivel process of tomatoes can be silenced or slowed down by a genetic modification. The next block of the picture shows how this process could be delayed by modifying the genes. This modification let the tomato consumers to keep them for a longer period of time. Similarly, the rightmost picture shows the difference between the domestic papaya and genetically modified papaya, which is resistant to a common viral disease. Let's learn some basic of biology. Have you seen this picture before? If you can remember the things that you learned in your school science class, yes. This is an image of a cell, which is the building block of life. It is the smallest unit of life. An understanding of cells is therefore vital in any understanding of life itself. All organisms are made up of one or more cells, and all the life functions of an organism occur within cells. Moreover, cells come from pre-existing cells. This means a life begins with a single cell and reproduce itself so that the number of cells increases and the development proceeds. There are two types of cells, prokaryotes and eukaryotes. Prokaryotic cells are usually single-celled and smaller than eukaryotic cells. Eukaryotic cells are usually found in multicellular organisms, but there are some single-celled eukaryotes. Can you name a single-cell organism? Also, can you name multicellular organisms? Take some time to find the answer to the above questions. Although cells are diverse, all cells have certain parts in common. The parts include a plasma membrane, cytoplasm, ribosomes, and DNA. Cytoplasm is a jelly-like thick solution that fills each cell and is enclosed by the cell membrane. It is mainly composed of water, lipids, salts, and proteins. The cytoplasm contains all of the organelles. Among such organelles are the mitochondria, which are the sites of energy production. Mitochondria are known as the powerhouses of a cell. They generate most of the chemical energy needed to power the cell's biochemical reactions. The purple color circle within the cell is called the nucleus. It is the central processing unit of the cell. The nucleus is a highly specialized organelle that serves as the information processing and administrative center of the cell. This organelle has two major functions. It stores the cell's hereditary material, or DNA, and it coordinates the cell's activities, which include the growth of an organism. A lysosome is a membrane-bound cell organelle that contains digestive enzymes. Lysosomes are involved with various cell processes. Other than the organelles that we discussed, there are several others as well. Now we will be looking at the content of the cell nucleus. The content of the nucleus is packaged into a fiber, a thread-like structure resulting a fiber known as chromatin. Imagine a bowl of steamed noodles. This fiber is looped and coiled yet again, leading finally to familiar shape of chromosomes. Chromosomes are not always present, only when the nucleus is about to divide. The next slide will give you a better idea about how these are organized within the cell nucleus. As I mentioned earlier, the noodles-like thing, the chromatin, is a loose combination of DNA and proteins. The chromosomes are made up of a pair of chromatids. Achromatids are the two thread-like strands into which a chromosome divides during cell division. Each contains a double helix of DNA. Chromosomes are not visible in the cell's nucleus. Not even under a microscope when the cell is not dividing. However, the DNA that makes up chromosomes becomes more tightly packed during cell division and is then visible under a microscope. Most of what researchers know about chromosomes was learned by observing chromosomes during cell division. Chromosomes carry genes which hold the information to build and maintain an organism's cell and pass genetic traits to offspring. How many chromosomes do human beings have? In humans, each cell normally contains 23 pairs of chromosomes, for a total of 46. 22 of these pairs, called autosomes, look the same in both males and females. The 23rd pair, the sex chromosomes, differ between males and females. Females have 2 copies of the X chromosome, while males have one X and one Y chromosome. In fact, each species of plants and animals has a set number of chromosomes. A fruit fly, for example, has 4 pairs of chromosomes, while a rice plant has 12 and a dog, 39. Inside the nucleus, DNA molecules can be found. DNA stands for deoxyribonucleic acid. It is the hereditary material in humans and almost all other organisms. DNA is made up of molecules called nucleotides. Each nucleotide contains a phosphate group, a sugar group and a nitrogen base. The 4 types of nitrogen bases are adenine, A, thymine, T, guanine, G, and cytosine, C. The order, or sequence, of these bases determines the information available for building and maintaining an organism, similar to the way in which letters of the alphabet appear in a certain order to form words and sentences. DNA bases pair up with each other, A with T and C with G, to form units called base pairs. Each base is also attached to a sugar molecule and a phosphate molecule. Together, a base, sugar, and phosphate are called a nucleotide. Nucleotides are arranged in two long strands that form a spiral called a double helix. The structure of the double helix is somewhat like a ladder, with the base pairs forming the ladder's rungs and the sugar and phosphate molecules forming the vertical side pieces of the ladder. If any single DNA molecule is stretched, it would be quite long. For an example, human DNA molecule can be stretched to 5 to 6 feet. To get them packed within the nucleus, these are extremely condensed. Gene, unit of hereditary information that occupies a fixed position, locus, on a chromosome. Genes achieve their effects by directing the synthesis of proteins. Genes forms as few stretches of DNA that contains genetic instructions used in development and functioning of all living organisms. These genes codes for traits that are physical characteristics of an organism. Every person has 2 copies of each gene, one inherited from each parent. Most genes are the same in all people, but a small number of genes, less than 1% of the total, are slightly different between people. Alleles are forms of the same gene with small differences in their sequence of DNA bases. These small differences contribute to each person's unique physical features. Some alleles are dominant or recessive. When an organism is heterozygous, different, at a specific locus and carries one dominant and one recessive allele, the organism will express the dominant phenotype. For an example, let's consider the pair of alleles that code for the skin color. Also, assume that there are only 2 colors, namely, black and brown. If black is the dominant allele, whenever at least one dominant allele present in the pair of alleles in an organism, the skin color of that particular organism will be black. The only instance that the skin color is brown will be when the pair of alleles contains 2 recessive alleles. A letter in the English alphabet can be used to denote an allele. In general, an uppercase letter is used for a dominant allele and lowercase letter is used to represent the recessive allele. A genome is an organism's complete set of DNA, including all of its genes. Each genome contains all of the information needed to build and maintain that organism. In humans, a copy of the entire genome, more than 3 billion DNA base pairs, is contained in all cells that have a nucleus. If the instructions in a human genome is to be stored, it will accommodate 700 megabytes. How this human genome instructions were decoded? The Human Genome Project was an international research effort to determine the sequence of the human genome and identify the genes that it contains. It formally began in 1990 and was completed in 2003, 2 years ahead of its original schedule. Now, you should have an idea about how complex is this task of decoding the instructions of an organism's genetic information. For an example, let us see the content of a chromosome of a pea plant. Here, I have shown 7 pairs of alleles that code for different genes. You may visit the following link to see how the cell division is taken place. The genetic makeup of the trait is called the genotype while the physical appearance is called as the phenotype. That is, the genotype codes for its phenotype. Let's look at a classic example, eye color. A gene encodes eye color. In this example, the allele is either brown, or blue, with one inherited from the mother, and the other inherited from the father. The brown allele is dominant, B, and the blue allele is recessive, B. If the child inherits two different alleles, heterozygous, then they will have brown eyes. For the child to have blue eyes, they must be homozygous for the blue eye allele. Before we move into the next section, one last thing that I would like to introduce is the difference homozygous and heterozygous alleles. Homozygous means two copies of the same allele, such as two dominant alleles. Heterozygous means one of each type of allele, one dominant and one recessive. This ends the very brief introduction to basic biology. Though this is not a biology class, I strongly suggest you to refer additional materials to get a good idea about these concepts. In the next section, we are going to talk about the Mendel's experiment.